So today our guest is Dr. Srini Ramasamy. I always read the name very well, but every time I have to search it online, I wonder whether it is Ramisamy or Ramasamy for some reason. But Dr. Srini is the Interim Deputy Director General, Partnerships and Flagship Program Leader, Safe and Sustainable Value Chains, and Lead Entomologist at World Vegetable Center in Taiwan. So Dr. Srini received his PhD in agricultural entomology from Tamil Nadu Agricultural University in India, same university probably with Dr. Subi. And he also worked with that university in partnership with Asian Vegetable Research and Development Center, Taiwan, in 2003. Then since 2005, he has served as a lead entomologist at the World Vegetable Center and is currently leading the flagship program for safe and sustainable value chains. He also served as a member of the World Veg Research Management Team. Dr. Srini is an agricultural entomologist specializing in host plant resistance, biological control, chemical ecology, in insect pests, molecular entomology, and integrated pest management. He has obtained over 31 million USD research and development projects, and has the project and as a project manager, flagship program leader, and acting regional director. He has coordinated research and the formation of linkage among more than 30 countries in 6 continents. Partners, including universities, private sectors, farmer groups, NGO, and governmental and international organizations, have also been a part of his linkage. He has supervised and trained 75 students, postdoctoral fellows, intern research technicians, and associates from around 15 countries. He has published over 225 papers, including research and review articles, book chapters, field guides, and conference proceedings. One of his notable publications is a review article on Maruca vitrata in the Annual Review of Entomology published in 2021. He's received several awards, including the International Plant Protection Award of Distinction from International Association for Plant Protection Science in 2011, the ISHS Medal from International Society for Horticultural Science in 2018, Veginet CVEG 2019 award during Southeast Asia Vegetable Symposium, and Fellow of the Royal Entomological Society, London in 2021. Countries of work experience include Australia, Bangladesh, Benin, Cambodia, Cameroon, China, Colombia, Eswatini, Ethiopia, France, Germany, Hong Kong, Indonesia, Italy, Kenya, Laos, Malaysia, Mongolia, Myanmar, Nepal, Netherlands, Papua, Guinea, Philippines, Singapore, South Africa, Korea, Sri Lanka, Taiwan, Tajikistan, Tanzania, Thailand, Turkey, United Kingdom, United States, Uzbekistan, and Vietnam. So, Dr. Srini, welcome once again. So you have the floor. So the topic today is titled, "Are we winning the tug of war with the diamondback moth?" So Dr. Srini, please, you have the floor.
Thank you, Salio. Can you see my screen?
Yes.
Okay. Okay. Good morning, good afternoon, or good evening from wherever you are joining. First of all, I would like to thank Salio, my good friend, and also AAIS for inviting me to share this knowledge. I have been working with many colleagues in ISIPE, in IITA, and it's a pleasure to be associating with the AAIS at this moment. So when Salia was inviting me to give a webinar, I was asking myself what should be the topic. And Salio introduced me. One of my most favorite insects is Maruca vitrata legume pod borer, which is quite prevalent in both Asia and Africa. So I thought, should I talk about the Maruca vitrata? But then the other insect with which the World Vegetable Center has been working for almost for the past 4 decades, even before my joining, is diamondback moth. And like a couple of months ago, I was in West Africa. I was traveling in the vegetable production regions in Benin. Then I again encountered a very severe infestation of diamondback moth and other caterpillar pests on brassicas in crops such as cabbage. So I thought of sharing this particular topic on diamondback moth because there are a lot of opportunities to exchange knowledge and also the technological innovations between Asia and Africa to manage the insects. So in this webinar, I will be sharing some of our past work and how we exchanged the collaboration between Asia and Africa to manage this insect. And are we able to successfully manage diamondback moth and other insect pests on brassicas? Okay. So needless to say, the brassicas are very important crop, both for the income and also for the dietary diversity and the nutritional importance. And it's very diverse. When we say the brassica group, it ranges from the leafy vegetables to even root vegetables like radish. So this is an example from Southeast Asia, where they cultivate several kinds of leafy brassicas. In fact, some of the crops are very short duration crops, which can be harvested in a span of 45 to 60 days, and they generate a huge income. So you can see here in countries like Cambodia, people are able to generate like $7,000 per hectare just in a cropping cycle. So it's a similar scenario in most countries, even in Africa. Crops like kale and cabbage are very important crops. So, but one of the most important constraints in the production of brassica vegetables is diamondback moth. Of course, it's cosmopolitan, and it can occur in any kind of weather condition, in any kind of altitude. So the diamondback moth is the most serious problem. And as you see in this particular photograph, you can see so many caterpillars feeding on a single leaf. And if the crop is left unmanaged, then you can harvest nothing from the field. So a complete crop failure is a possibility, as shown in this photo. So in most places, no matter where the brassica crop is grown, by a large scale commercial farm or by a smallholder farmer, they tend to spray a lot of chemical pesticides to manage the diamondback moth. And if you walk through any intensive vegetable production regions in Asia or Africa, this is a common scenario. The farmers tend to use a lot of chemical pesticides, most of the time the cocktails. And we have a study from Southeast Asia, which documented the farmers spray like twice a week, and then they spray 3 to 4 compounds in single spray. So it's such an intensive and indiscriminate use of pesticides. So a conservative estimate Of course, it was done in like 12 years ago. The figures could be very different now. A conservative estimate covering both the damage and losses due to the brassica production and the management cost for the diamondback moth alone is around 4 to 5 billion US dollar worldwide. So you can just imagine the magnitude and importance of this particular insect pest. Okay, at this juncture, I would like to draw your attention to the life cycle of diamondback moth. This is the egg stage. And you know, the eggs are just laid on the leaf surface and even on the upper leaf surface in clusters. And then when the caterpillars are hatched out, they feed on the foliage. And when it becomes pupa, it's pupating in a very loosely woven cocoon. And again, the pupation happens on the leaf surfaces or on the plant surfaces. And then the adult moths come out, and it's a very tiny moth and a weak flyer. So here you might have noticed the egg, larva, pupa, adult, all are present on the leaf surfaces. It means they are highly exposed to the natural mortality factors, be it a abiotic factor or the biotic factors like the natural enemies or the entomopathogens. Despite the presence and the vulnerability of the life stages of diamondback moth, still, this has assumed the primary pest role in the brassica production system. So this is an amazing fact for this tiny moth to survive and dominate the brassica production systems until today. Okay, so when we wanted to control this particular insect pest, so World Wage was taking a leading role. Of course, my predecessor, Dr. Talekar, some of you might be knowing him, took an initiative in early 1980s to introduce this particular larval parasitoid, which is called the Diadigma semiclasm, which is highly adapted to the highlands or to the lowlands in the cooler seasons, wherever the temperature is less than like 32 degrees centigrade. Okay, so if we look at the history of the Diadigma semiclasm, the Diadigma semiclasm originated in UK, and then it was introduced in 1936 to New Zealand, because you know, New Zealand produces lots of forage brassicas, and diamondback moth is a #1 problem. And since this is a forage brassica, the farmers are unable to spray any chemical pesticide. So they relied on the introduction of Diadigma semiclasm to control the diamondback moth, so that they can produce a good biomass to be used as a forage crop for their cattle industry. So upon seeing the success, the Diadigma semiclasm was introduced from New Zealand to Indonesia in 1950s. But in Indonesia, it was the time where the broad spectrum pesticides were started to be used in the agriculture industry. So because of the extensive use of the broad spectrum pesticides, they not only wiped out diamondback moth, but also wiped out the Diadigma semiclasm. So it took another 20 years in Indonesia until they start using the Bacillus thuringiensis based biopesticide. The Diadigma semiclasm was unable to establish in the brassica production systems. But since 1970s, some of the farmers started using the BT biopesticide. The Diadigma semiclasm established quite well. And then we started realizing the potential of this parasitoid. So subsequently, Dr. Talekar from World Vegetable Center, at that point of time, it was known as Asian Vegetable Research and Development Center, or AVRDC, got this parasitoid, Diadema semiclassum, from Indonesia and introduced it into Taiwan, where World Wedge headquarters is located. And upon completing all the biosafety requirements and the regulatory requirements, these parasitoids were introduced in the central highland regions and the northern highland regions of Taiwan, where the brassica production dominated at the time. And within a period of like 2 to 3 years, the parasitoid established and started spreading in most parts of Taiwan, because this is a tiny island nation. So it started establishing and spreading itself. So this showed the potential of this parasitoid to be subsequently introduced into many countries. So since 1989 to 1996, this was introduced into a couple of countries in Southeast Asia like Malaysia, Indonesia, Philippines. And then subsequently during 1996 to 2005, it was expanded to countries like Cambodia, Laos, and Vietnam. And at the same time, in 1992 to 2000, it was also introduced into the South Asian countries, including India, Pakistan, Sri Lanka, Nepal, and Bangladesh, all those countries. So in a period of like 20-25 years, the Diadema semiclassum was introduced into almost all the countries, starting from East Asia, Southeast Asia, and South Asia. So in the beginning of this century, upon realizing the potential of this parasitoid, Thalekar, along with Dr. Bernard Lohr, who was an entomologist at ISIPE at the time, they took a joint project, and then they started introducing this Diadema semiclassum into the Eastern and Southern African countries during 2004 to 2008. So in East Africa, the predominant caterpillar pest is diamondback moth, besides aphids and Baghdad beetle.
Sorry, it seems that your slides are not moving, or is it the same?
It's the same. It's the same.
Okay.
Yeah. So then with the introduction of Diadema semiclassum, it started reducing the impact of diamondback moth in Eastern and Southern African countries during 2004 to 2008. Okay. But you are well aware that any parasitoid in a natural environment can exert a parasitism of like 40 to 50% a maximum. It cannot completely control a particular pest. So this is the same with the Diadema semiclassum. It attacks the second and third instar stage, and it controls most of the caterpillars of diamondback moth. But still, like half of the caterpillars are successfully becoming pupa. So we wanted to identify a parasitoid, especially a pupal parasitoid, that could complement the performance of Diadema semiclassum in the highlands. So this is where we identified the candidate Diadromus scholaris, a pupal parasitoid, again well adapted to highlands or to lowlands, particularly during the cooler season. And both Diadema semiclassum and Diadromus scholaris were able to control almost like 70 to 80% of the diamondback moth population. And this is a very classic example of the classical biological control program in Asia. Okay, so based on the progress and success over a period of like 15 years, both Dr. Thalekar from AVRDC, or the World Vegetable Center, and Dr. Anthony Shelton, or Tony Shelton, from the entomology department of the Cornell University, particularly the New York State Agriculture Experimental Station in Geneva, they both put together all the progress into a review paper, which got published in the year 1993. So this was the first annual review on the comprehensive biology, ecology, and management of the diamondback moth. And then in Asia, some of the impact assessment studies were carried out, because we wanted to see what was the reduction in terms of the use of insecticide applications in brassica production systems. So the studies revealed that in countries like Indonesia, the pesticide use was reduced by 50%. And in Malaysian highlands, especially the Cameroon highlands, which is a major hub of cabbage production in Southeast Asia, the pesticide reduction was even up to 86%, and in the Philippines, it was 60%. But it's not just the reduction in the chemical pesticides or reduction in the cost of plant protection or increase the income, but even when we eliminate a heavy load of chemical pesticides from the ecosystem, the whole biodiversity in the production system start increasing. So a study from Indonesia documented that it has a huge impact on the soil biodiversity. For example, some of the soil dwelling predators, they increased by 84% in Indonesia. Okay, so then we wanted to share the success and the knowledge with the outside world. So we wanted to bring together all the experts working on the diamondback moth into a network. And then this informal group started organizing an international conference or international workshop, they call it at the time. So the first one was organized in AVRDC or World Wage in Taiwan in the year 1985. And then subsequently, the second one was also organized in Taiwan. And then it was moved to the different countries in the Asia Pacific region, like the third one in Malaysia, fourth in Australia, fifth one in China. But if you notice from the third one onwards, it was not only focusing the diamondback moth, but also looking at the other brassica pests. So I will come to the reason why in a moment. But this is how the conference is being organized over a period of last 40 years. So we organized up to 9 conferences. The 9th one was organized in Cambodia last year. And now it has become a regular event once in 4 years. And the 10th conference is scheduled to be organized in Africa. So most likely it will be organized in East Africa in the year 2027. And still, this is a kind of like informal working group focusing on the diamondback moth and other brassica pests. And the working group is managed by World Wage. And within the World Wage website, there is a site for the working group, which also contains all the conference proceedings up to the 8th one, and the 9th one is in progress, which will be published in a month or 2. So these are all openly and publicly accessible. Anybody can access all the papers. Okay, so I could end the story here, but the story doesn't stop here. The reason is, if you have carefully noticed, most of the time I was using the word highlands, cool seasons, etc. So it means still we have some problems in the lowland brassica production systems, because the parasita like Diadigma semiclassum could not perform well in a temperature regime of over like 33 degrees centigrade. And at that point of time, most of the progenies will be the male, and it naturally destabilizes the population. So it doesn't adapt to the high temperature condition or to the lowland brassica production system. So when we were looking for some equivalent and competent parasitide fitting for the lowland brassica production systems, then we ended up getting the Cotesia plutelle, which is now known as Cotesia vestalis. And again, Cotesia vestalis, like Diadigma semiclassum, is a larval parasitide controlling diamondback moth quite well in the lowland brassica production systems. But as I said before, the parasitide could exert a natural parasitism of about 40% or 45 or 50% in the field conditions. It means still in the lowland brassica production systems, half of the population is successfully becoming pupa. So here, again, following the success case of Diadromus scholaris, a pupal parasitide for the highland production systems, we wanted to find an equivalent pupal parasitide for the lowland brassica production systems. Cotesia vestalis is very commonly found in most Asian countries and also in some of the African countries like South Africa, you can find Cotesia vestalis. But we were unable to find any pupal parasitoid that successfully adapted to the lowland conditions for a high temperature regime in Asia or even in Africa. So then subsequently, it has become a major constraint for the successful biological control program of diamondback moth in the lowland production systems. So in the beginning of this century, some of the literature started coming out, especially from the Mediterranean regions in countries like Romania or Syria, reporting the presence of a wide array of parasitoids in those countries for diamondback moth. So it has become a very interesting region because in those regions, the temperature could easily hit like 40 or even 45 degrees centigrade. And the brassica production is a bit common in the lowland production systems. So with a hope to find a pupal parasitoid, I made an exploration back in Syria almost 20 years ago. So I went on a mission for almost like 3 successive years. And I visited different locations like in Aleppo, Deir ez-Zor, Homs in the central Syria, and Damascus. And over a period of 2 years, I was able to find a candidate parasitoid, which is occurring in a very high frequency in the lowland brassica production systems. And the fact is, it's not a pupal parasitoid, it's a larval parasitoid. And then once we got the adults, again, I was really shocked because it's none other the Diadema semiclassum. It was really a surprising outcome because until then we were believing that Diadema semiclassum was adapted to only the highland situation or to the low temperature regime. But this particular strain, morphologically, there was no difference. And then we even performed the molecular comparison, there was no difference. So then we compared the population coming from Syria and the population existing in Taiwan, which originally came from UK. So we did a wide exposure, temperature exposure experiments, and the Syrian strain adapted and performed much better in a range of 15 to 35 degrees centigrade. And even at the highest temperature of like 35 degrees centigrade, you can get like 25% parasitism, where Taiwan strain almost declined to a level of 6%. So this is an interesting strain coming from Syria, adapted to a very high temperature regime. But all these experiments were carried out in the quarantine condition. And then when we are ready to get the approval from the authorities, because of the escalating situation in the Syria, I was unable to go back and get this particular strain. But this particular strain is an interesting strain to be considered for the lowland brassica production system. But still, this particular strain does not provide us a solution because already we have Cotesia plutelli or Cotesia vestalis adapted to the lowland conditions. So still, we are looking for a pupal parasitoid, not a larval parasitoid for the lowland brassica production systems. Okay, so now you hear the story for the highland system and for the lowland system. So now you might have a question. Okay, at least for the Asian highland system, are we satisfied with the diamondback moth biological control? Yes. We are quite convinced that we could manage the diamondback moth with the introduced candidate parasitoids, Diadema semiclassum and Diadema scholaris. But what happens the moment you take away diamondback moth from the production system? You get all the secondary lepidopterans and the secondary coleopterans dominating the production system, growing and occupying the level of the primary pest, like the Pieris, cabbage white or cabbage butterfly, cabbage cluster caterpillar, which is Crocidolomia pavonona, cabbage webworm, Helula undalis, and striped flea beetle, which is the Philotrita species. And if I remember correctly, In Africa, besides diamondback moth, you also have this cabbage cluster caterpillar, Crocidolomia, and cabbage webworm. And in fact, cabbage webworm at times even occupies the primary pest role in Central and West African region. Okay, so then we thought, okay, can we provide a comprehensive solution to most of the caterpillar pests? Because you have cabbage butterfly, diamondback moth, and cluster caterpillar, cabbage webworm, and even armyworm, sometimes helicoverpa. So we thought, okay, can we provide a comprehensive solution through the transgenic plant? Because at that point of time, there were studies at the Cornell University which showed, okay, you create a transgenic plant with one gene, like Cry1AC gene, and then expose the transgenic plant to diamondback moth. But you know, the diamondback moth within a 12 generation time period is able to overcome the transgenic resistance. And then at that point of time, he introduced another transgenic plant with the Cry1C gene. Then at least the Cry1C plant is able to suppress and maintain the susceptibility level for another 12 generations. Okay, so it means that when you deploy the transgenic plant with one gene, the insect will be able to overcome within a period of 12 generations. And also when you introduce this into a mosaic system, again for the individual genes, the insect can develop resistance fairly quickly, just like in 12 to 15 generations. But when you put two genes into the same cartridge and then develop a transgenic plant, and you can see the susceptibility level is maintained for a prolonged period of time. So this was the proof of concept. In the beginning of this century, we confirmed. And then we wanted to enter into a public-private partnership called SIMBA, Collaboration on Insect Management for Brassicas in Asia and Africa. And it brought together 4 public sector institutions, including Cornell University, Natural Resources Institute, University of Melbourne, World Wage, along with the Nunhem seed. And then we were targeting 2 genes, Cry1B, Cry1C, and we tested these toxins in different countries against diamondback moth, and the populations in different countries are very susceptible. And also we tested the secondary lepidopterans for both Cry1B and also Cry1C, and most secondary lepidopterans are also susceptible. But when we wanted to put the genes into the transgenic plants, you know, we were targeting the India as the first target country because of the existing advanced regulatory system for the introduction of the transgenic plant. We were working on this project, but when the plants are ready to be tested and deployed, there is an indefinite moratorium on the introduction of transgenic plants, especially the food crops in India, which actually forced us to stop this project. So at that point of time, I continued assessing the toxicity assays for at least like Cry1A genes and Cry1C genes. The reason is some of the commercially available BT formulations are based on Cry1A toxin or Cry1C toxins, like Zentari, which is based on the subspecies Isavai, BT Isavai, so which is having Cry1C, and all the BT Kurstaki based formulations are having Cry1A toxins. So at least we can deploy in a sequential manner both the commercial formulations. So we wanted to check both the diamondback moth and the secondary lepidopterans are susceptible, and we confirmed, and then we made sure at least we should be able to use the different commercial BT formulations for managing the diamondback moth. Okay, so now we have a solution for diamondback moth. We have the solution for the secondary lepidopterans. So then the another noxious problem, unmanageable problem, at least in Asia, is the flea beetle. And what you see here is called striped flea beetle because of the 2 golden stripes, which is the Phyllotritus triolata, which is a chrysomelid beetle. So when we were scratching our head what to do with this particular species, there are some existing information which are encouraging information that these flea beetles are producing aggregation pheromones. So the crucifer flea beetle, Phyllotritus cruciferae, which is common in Europe or in North Africa, and for other species like Euthona, they already identified aggregation pheromones. In this species, the males release the aggregation pheromone, which attract both sexes, both males and females, which is a kind of like sending out the information on the availability of the host plants. So based on this fact, we wanted to try to identify the aggregation pheromones in the striped flea beetle in Phyllotritus triolata. So in a collaboration with the Humboldt University Berlin and the Max Planck Institute for Chemical Ecology in Germany, eventually we were able to identify two active compounds that attract the conspecifics of the striped flea beetle, which is the Himachala 2,4-diene and another second compound. And even the field experiments and the lab experiments confirmed that when we combine the pheromone compound with the host plant volatile, probably you might be aware that the brassicas are having the compound called glucosinolates. And the glucosinolates, when they come into contact with the myrosinase enzyme, they are hydrolyzed and they become the low molecular weight volatile compounds, the isothiocyanates. So there are like allyl isothiocyanate, alkyl isothiocyanates, aryl isothiocyanates. So the alkyl isothiocyanates is one of the most active compounds, and it's also highly attractive to the specialist feeders like the flea beetle or diamondback moth or cluster caterpillar. So when you combine the allyl isothiocyanate with the aggregation pheromone compound, they are quite attractive. So at this point of time, we should have an active player, a private sector player who should be able to synthesize and develop the lures to be deployed against these insects. So this is where we are standing currently. But at the same time, we wanted to explore the other cultural aspects like using the trap crops. Probably you may be aware that the Indian mustard is a popular trap crop used against the diamondback moth in Asia and the Pacific region. And surprisingly, we found another plant, which is spider plant. The Cleome species was found to be attractive because the Cleome species is very closely related to Brassicas. In fact, the Cleome was earlier classified under the same family Brassicaceae, but now they have been moved to the separate family Cleomaceae, but they both are under the same Brassicales order. So the Cleome species, they are also having the glucosinolate compounds like the brassicas. And because of the presence of glucosinolates, especially during the off-season, the flea beetles or even diamondback moth or Crocidolomia, they move to feed on the Cleome species. So we wanted to check the available spider plant species within our gene bank, and we were assessing against a range of insects, including diamondback moth, Crocidolomia, Hellula, in comparison with the standard known trap crop like Indian mustard or pak choi. And then here you found spider plant is not very different from the known trap crops in terms of attracting these insects. And even the spider plant is able to attract the insects to dump a lot of eggs on them, but they don't sustain the growth of the caterpillar successfully. So it means these spider plants can be explored as the dead-end trap crops for the management of some of these insects, including the flea beetle. So in the field experiments, we were able to demonstrate that with an increasing proportion of the trap crop, the population of the diamondback moth on the main crop like brassicas can be significantly increased. But then if you notice, you need to have like at least like 16 or 20% of the crop should be under the trap crop. So unless the trap crop has some economic value, it is very difficult to convince the smallholder farmers to allocate 20% or 25% of their area for planting the trap crops. So this is a tricky area. So possibly, in some regions where the trap crops have some economic value can be considered for deployment. Okay. So when we wanted to complete the whole story, so you have like some cultural practices, you have the biopesticides, and you have the biocontrol. So we want to put them into your IPM package. And in a project in Southeast Asia, like Cambodia or Laos, we tested this IPM package along with the farmer's practice. The farmer's practice is simply the calendar-based application of chemical pesticide. So here you can see for most of the pest species, including the Plutella, Xylostella, diamondback moth, armyworm, Spodoptera litura, then the cabbage webworm, or even the flea beetle, the damage is almost the same or even much reduced in the IPM than the farmer's practice compared to the untreated control. And even the marketable yield is significantly increased compared to the untreated control. So this is quite an encouraging one. And then here I like to draw your attention to one particular aspect. Yeah, this is a result from a biopesticide trial. So we used the BT formulations like Zentari or Crimax. These are all like BT Izava or BT Kustaki. And the real M62 is the metarhizium. And this is coming from ISIPE and commercialized by the real IPM company. So they were part of this project. So we got this product from real IPM. So if you look at the results, particularly for the flea beetles, even for all the BT or the entomopathogenic fungi, the damage level was much lower, similar to the chemical pesticide compared to the untreated control. So here you might have a question. The BT formulations are active against the lepidopterans. How could they reduce the damage by the flea beetle, which is a coleopteran? So we went on experimenting this particular hypothesis because the only possibility here you need to correlate the chemical cues emanating from the glucosinolates that attracts the Brassica specialist feeders. So the hypothesis here, okay, you use the biopesticide, and then you control the caterpillar pests like diamondback moth or the other secondary caterpillars. So because you control the damage, plant damage due to diamondback moth or other caterpillars, the production of the allyl isothiocyanates or other kind of isothiocyanates is reduced, so the plants become less attractive to pests like flea beetles. So we wanted to test this hypothesis. So we set up different treatment like plant with the diamondback moth, but the plant with the diamondback moth and you spray the BT formulations or chemical pesticide. And even we had a treatment where we do not have any caterpillar pest, but you mechanically damage. Because even when you mechanically damage, you can bring together the glucosinolate and the myrosinase enzyme, which can produce the volatiles and attract the herbivores. So this is why we introduced a mechanical damage treatment, and there is a plant without any damage. So then you can see here clearly the plant with the diamondback moth or the plant mechanically damaged, they are having the more number of flea beetle damage. But when you treat the plants with the BT formulations or chemical pesticides, because of the reduced damage by the diamondback moth, the plants become less attractive to the flea beetles, and then the damage is much reduced by the flea beetles. So here, although this is not a direct impact of the biopesticide on the coleopteran pest, it's because of the trade-trophic interaction, the coleopteran damage is also eventually reduced. So here what I want to highlight, when you have a combination of technologies in the IPM package, due to the interaction, you will be able to suppress the lepidopteran, coleopteran, and the aphid pests on the Brassica production system. And to end the whole story, okay, you have the IPM and you deploy the IPM and you are able to suppress the pest population and you are able to increase the yield. But the other ecosystem services that we are actually getting out of the adoption of this IPM is the proliferation of the natural enemies in the production system. So these are the field evidences coming from Laos or Vietnam in Southeast Asia, where we deployed the IPM using the biopesticides and pheromones, then the natural proliferation of the parasitoids like Cotesia glomerata, which is a parasitoid of cabbage butterfly, or cabbage butterfly pupae, the Pteromalus puparum. These natural enemy populations started increasing gradually. And the same thing goes with the aphid parasitoids as well. So because of the adoption of the IPM based on the biopesticides and the natural enemies, over a period of time, we are able to build up the biodiversity in the production systems. And then eventually, even we can bring down the natural incidence of the pest populations below the economic threshold level. But it needs a community approach. It needs the cooperation among the farmers in the particular region. So area-wide approach is not only important for the technologies like pheromone, they are also quite important for the successful impacts of IPM technologies. Okay, to conclude, for successful control of diamondback moth, we should have a broad spectrum approach of focusing on all the pests, no matter aphids, secondary lepidopterans, the coleopteran pests. We need to have an approach that could tackle all the pests of the brassicas. And then we should combine the biocontrol, the classical biocontrol based on the introduced natural enemies, with other components of IPM, especially the biopesticides. So the latter component, the biopesticides, actually need a concerted efforts at the nationwide, because in most countries still there are like regulatory and policy barriers. Until recently, most countries in Southeast Asia, they do not have a regulatory system for the introduction of biopesticides. And only with the great effort of the BMZ through GAIZ in Southeast Asia, they harmonized the process, regulatory process for the commercialization of the biocontrol agents in the Southeast Asian region. And I'm sure this is the same concern existing in many countries in Africa. So there should be an effort to address the regulatory and policy barriers to introduce more biopesticide options for tackling the pests on brassicas or other vegetable crops. And then the sustainable adoption. Actually, we need to work with the farmers and encourage them to adopt these eco-friendly approaches. So we should find the potential entry points. For example, in Southeast Asia, recently they started introducing the good agricultural practices certified production systems, the GAP production systems. And these farmers are actually starving for the eco-friendly innovations to replace the use of chemical pesticides, because the certified production systems like GAP or organic certified production systems, they can't rely on the chemical pesticides. So they are looking for the potential solutions. So we should target these entry points so that we should be able to promote the level of adoption of IPM. And the most important thing is all these options should be available to the farmers at affordable prices and within the reach. Otherwise, the farmers will not be able to adopt the innovations like biopesticides. So this is where we need to work very closely with the private sector, because the public sector can do a lot of research. And we can address all the critical research questions on target or non-target effects, everything. But if we want to successfully commercialize the product, we need to have a strong involvement of the private sector, which was even evidenced in East Africa by the active participation of the private sector, which commercialized several IPM component technologies. So this is what you need at this moment in the continent-wide approach in Africa to take the IPM technologies to tackle diamondback moth or other associated lepidopteran or coleopteran pests, especially in Western Central Africa region. Compared to Eastern Southern Africa region, still Western Central Africa region is having very few options to tackle this particular insect. And the farmers are extensively relying on the chemical pesticides, which is posing a lot of threats, including the human and environmental health safety threats. So with this, I would like to conclude my presentation, and I will be glad to answer any questions that you have. Thank you very much.
Thank you very much, Dr. Srini. Excellent presentation.